Okay. Please confirm whether my screen is visible or not. Is it visible?
Yes, sir.
Okay. Okay then. Let's start the meeting. Hmm. Welcome everyone. So we are now in week 9. So it is bit like earlier we have started this module, like the discussion session. Actually, due to some unforeseen reasons, I wanted to conduct it, prepone this meeting. So week 9 is about membrane protein and transport. In this module, you will learn about the different transport membrane protein and the thermodynamics of the membrane protein interaction, then electron transport chain, and many more things that are very much interesting in research perspective. And also, these are the main target of any competitive exam. So whether you are writing GATE, NET, or any exam which involve the biochemistry, protein chemistry, or thermodynamics, means biochemical thermodynamics, you will find the questions from this module. So it will be very much like kind of helpful for you you if you keep more focus on the this module in your fundamentals of protein chemistry course. So the concept that will be covered in this module, because I assume that most of because the this module was released yesterday only, so I feel that most of you have not gone through the lectures, but still I would like to cover few of the concept, and then I would like to take some sample problems from this module so that you can have the handy, like you can have the confidence while attempting the question in assignment, also in the uh, competitive exams. So the concept that will be covered in this week, by mistake it is written at week 8. Let me correct it to week 9. Okay. So in the week 9, the concept that will be covered are membrane lipid distribution, then lipid bilayer, then types of membrane proteins, then transmembrane region and polarity of this, then membrane structure and potential, transport proteins that are available in the transmembrane like lipid bilayer, then the electron transport chain, which consists of the complexes such as from complex 1 to complex 4. There is a specific name for all the complexes. Then you will be learning about the thermodynamics of membrane protein and electron transport chain, which are electron transport chain are available in the mitochondrial cristae, the inner mitochondrial matrix membrane. Then the factors affecting the oxidative phosphorylation. Then the important keywords for this are the phospholipids, sphingolipids, sterols. These all are attached to the transmembrane. Like these all are the moieties that have been found on the lipid bilayer. Then the membrane proteins such as alpha helical membrane topology, then beta sheet helic topology that you found in most of the lipid bilayer. Then ATP synthase, which is the main machinery to synthesize ATP. Then cytochrome ABC, ubiquinone, heme ABC, then mitochondrial aerobic respiration, because mitochondria is the powerhouse of cell. You must have known that in your like earlier classes and from your like basic level biochemistry or biology in 10th standard also. You must have incorporated the term that the mitochondria is the powerhouse of the cell. The main reason why it is called the why mitochondria is called the powerhouse of the cell, that you will learn in this module. Then glycolysis, the complex 1 to 4, then uniport, symport, and Na plus ATPase. These kind of terminology you will come across in this module. So the as previous as per the previous all the sessions that I have conducted, some sample problems will be given. Then the discussion on important topics, then doubts will be taken if you have any, and then scope of this module with research perspective. Now coming to the question, your first question is integral proteins are known as uh, option A, glycosylated proteins, lipid, then bilayer proteins, transmembrane proteins, or intrinsic proteins. What is your take on this question? Integral proteins are known as
Transmembrane proteins.
transmembrane proteins. Very good. Option C is correct. Why integral proteins are called as transmembrane proteins? Because when you visualize the structure of lipid bilayer, means you have the hydrophilic head and hydrophobic tail. Then like this, you have that lipid bilayer, like hydrophobic tails come in contact with them, and the hydrophilic head makes the contact with the extracellular or intracellular region, which are mostly composed, which are mostly in contact with the water. Okay, you can remember that the concept which is the like dissolve like, right? So in that, the integral proteins are those proteins which which are present across the membrane, which have the both hydrophilic region, hydrophobic region, and again hydrophilic region. There are three regions are there. So because they travel across the, like they are present across the membrane, so that is why they are called the transmembrane proteins. And other, what are the opposite of the integral proteins? Can you recall? Other than the, what is the opposite terminology of the transmembrane protein, which are found on the surface, either of the surface of the lipid bilayer?
Peripheral.
Very good. Those are called the peripheral proteins. Very good. The next question is sphingomyelin usually consists of option A, phosphocholine, option B, monosaccharide residue, option C, oligosaccharide residue, option D, sialic acid. Sphingomyelin. There are different type of naming convention according to the, like the in the lipid bilayer, the main, the monomer of that, it has the like in the two hydrophobic tail, they have the different composition, and the head also have the different composition. Based on that, you name the that particular monomer as with respect to their the ligand that present in that. So what do you call sphingomyelin? Sphingomyelin is made of which of the like residue? Is it is it phosphocholine or monosaccharide residue or oligosaccharide residue or sialic acid? What do you find in sphingomyelin?
Option A.
Very good. Those are phosphocholine. Then the next question is, which among the following is or are the function of plasma membrane? So why the all most of the organism have the plasma membrane, which is lipid bilayer? The main role of them, the statement one is regulate the fluidity over the membrane, fluidity of overall the membrane, to protect the cell interior from the environment. Statement 3 is selective export import of the molecules, ions and gas. Options are only 1 and 2 are correct. Option B, only 1, 2 and 3 all are correct. Option C, 2 and 3 is correct. And option D, 1 and 2 only are correct.
Option B.
Option B, all three are correct. Very good. Because plasma membrane is used, the main role of the plasma membrane is to separate the two different extracellular and intracellular region, right? So, and also the transport protein that are present in the plasma membrane, they selectively export or import the molecules and gas from exterior to interior or interior to exterior, right? To protect the cell interior from the environment. This is also correct because the if plasma membrane is rupture, that means the inner content of the cell will be exposed to the outer environment and the cell will burst. Thus, hence it is called the cell death. So it is to protect the cell interior from the environment and also to regulate the fluidity over the overall membrane. So option B is correct for this question. Now the next question is, transmembrane protein are composed of dash amino acid. which interact with dash group of membrane phospholipids. Option A stands for hydrophilic, B for fatty acyl, A for hydrophobic, B for fatty acyl, A for hydrophilic, B for fatty acid, A for hydrophobic, B for fatty acid. What are A and B in this statement?
Option B.
Option B means transmembrane proteins are composed of hydrophobic amino acids which interacts with groups of membrane phospholipid. Are you sure about this?
The fatty acyl B so we interacts with the fatty acyl groups of the membrane phospholipids. The
Very
hydrophobic.
good. Yes. Option B is correct. So the transmembrane protein are composed of the hydrophobic amino acid which interact with the fatty acyl groups of the membrane phospholipids. Now the next question is, which among the following is not true about the mechanism of transport through the cell membrane? Option A, passive transport is a spontaneous process. Option B, active transport requires energy for transportation. Option C, passive transport is a unidirectional process. Option D, active transport occurs against the electrochemical gradient. Which among the statement is not true?
So option C.
Why do you think so?
So because it is based on the passive transport is based on movement from according to the concentration gradient from higher to lower. So if the direction changes, the transport will also change.
Very good. Yes. And what about the other statements? Passive transport is a spontaneous process.
Yes, sir.
And active transports require energy of transportation. Good. Then active transport occurs against the electrochemical gradient because of this purpose only, because it is operating against the electrochemical gradient. That is why they need the energy to transport the molecule from like against the electrochemical gradient. That is why it is called the active transport, right? They use the ATP or other source of energy to transport from lower to higher, right? Or the against electrochemical gradient, right? So option, which one? Option C is correct, right? Passive transport is a unidirectional process is a wrong statement. Now, next question. All aerobic organism, okay? Aerobic means they require the oxygen for their respiration. All aerobic organism requires A to produce the basic energy of generating ATP, which is adenosine triphosphate, and ATP synthesis enzyme called P, though the process is called C, transport. Okay, your options are A is for synthase, C for electron. A stands for oxidative phosphorylation, B for ATPase, C for electron. A for oxidative phosphorylation, B for ATP synthase, C for ion, and A for oxidative phosphorylation, B for ATPase, and C for ions. So which one is the correct among four?
Option A.
Option A. The aerobic organism require oxidative phosphorylation to produce the basic energy generating by ATP by an ATP synthesis enzyme called the ATP synthase, and through the process is called the electron transport. Very good. The option A is correct. Now, the next question is, electron is transported through complex 2 to complex 3 in cytochrome C after by which mechanism? Option A is Q cycle mechanism. Option B is M cycle mechanism. Option C is TCA cycle mechanism. Option D is Kelvin cycle mechanism.
TCS cycle mechanism.
TCA cycle mechanism. Are you sure about it? From complex 2 to complex 3. What are these complex 1, 2, 3, 4?
Exactly name is not right now I can't recall.
Okay. So what is the correct answer regarding to TCA cycle mechanism? Any other answer I can get? What happens in TCA cycle? Can you recall what happens in TCA cycle? Can you tell me?
So in PCA cycle, the pyruvate is enter the tricarboxylic acid cycle to generate the CO2 and other all necessary byproducts. And
Hmm.
yes, that's
Okay. TCA stands for the tricarboxylic acid cycle. That is correct. Yeah, but TCA. Can you recall where the TCA happens? Like, what is the location of TCA?
TCA is in the lumen of the mitochondria.
Lumen of the mitochondria means like inside the
Inside
cristae,
the
right?
mitochondria. Yes, sir.
Yeah, inside the TCA. Okay. And where the complex 2 and complex 3 lies?
They are in the inner membrane of the mitochondria.
Yes, they are in the inner membrane of the matrix. So mitochondria. So here what happens there? Like in the matrix when TC cycle happens, they generate the NADH and CO2, right? So in that case, what happens when the electron need to be transport and that electron create a electrochemical gradient, and also the protons that are released, they create a like concentration gradient across the membrane in the intermembrane space of mitochondria and inside the cristae, like inside the inner membrane matrix. So in the inner membrane, complex 2 and complex 3 are found, where they the complex in between the complex 2 and complex 3, a specific type of enzyme, a specific type of macromolecule that are present. Those are called the QCO. Okay, they absorb the protons and electrons and they get reduced. They form the QH2. That mechanism you will find in the slides that ma'am has discussed it clearly. Also, I will show you some animation video similar to the video that I showed in the last discussion session. So I will show you how these complex 1 to complex 4 are being cooperative, and then they inculcate the ATP synthesis mechanism and they form the gradient, and using the ATP gradient they harness this energy and synthesize ATP. The specific location and all the cofactors that are involved in that, everything will be covered in that particular animation. So the correct answer here is Q cycle mechanism. Okay. Other cycles are Kelvin cycle is related to the like dark cycle mechanism, right? TCA cycle is for the oxy. These are for generating the to harness the pyruvate and use it to complete oxidation of the pyruvate, right? But the Q cycle mechanism is related to the oxidative phosphorylation. Okay, which happens in the electron transport chain, and complex 2, complex 3, complex 4, these all are the part of the electron transport chain. Okay, now the next question is, in the electron transport chain, the NADH dehydrogenase complex is termed as which complex? The which of the complex is called as NADH dehydrogenase? You have 4 complexes from 1 to 4. So your options are complex 4, complex 2, complex 3, complex 1, complex 2. Which is called the NADH dehydrogenase? Which complex? It is complex 1. There are all 4 complexes have a particular name based on the enzyme, the main and leading enzyme that are present in that. Because the complexes are nothing but the these are the association of the different enzyme, as the name defines NADH dehydrogenase. Okay. So here the main role will play the NADH. The electrons will be taken from the NADH. NADH is a electron acceptor, right? They termed as the electron acceptor molecule, and the end electron acceptor in a eukaryote aerobic organisms are nothing but the oxygen, right? But they get the electrons from different sources such as NADH, FADH2, and all the complexes are dumped based on the major enzymes that are involving in that particular complex. So complex one is basically nothing but the complex one, NADH dehydrogenase. Okay. So your next question is the difference between the membrane carrier protein and membrane channel protein. Okay. It is about the membrane carrier protein and membrane channel protein. Options are channel protein can mediate active transport, but carrier proteins cannot. Option B, channel protein are synthesized by bound ribosome, but carrier proteins are synthesized by free ribosome. Option C, carrier proteins are glycoproteins and channel proteins are lipoproteins. Option D, carrier proteins can mediate active transport, but channel proteins cannot. So, any idea about the membrane carrier protein and membrane channel proteins? What is the correct statement? The difference between the membrane carrier protein and membrane channel proteins? Any guess? The main difference between the membrane carrier protein and membrane channel protein are that the carrier proteins can mediate the active transport. However, the channel proteins, as the name specify the channel, that means they will only operate the passive transport. Okay, channels. However, the carrier proteins can mediate the active transport. So option D is correct. That is the basic or major difference between the carrier proteins and channel proteins. Okay. Your next question is in the complex 2 electron transport pathway, electrons are delivered from dash to the dash pool via dash that is reduced. Your options are like electrons are delivered from succinate to the quinone pool via flavin adenine dinucleotide or FES cluster quinone flavin adenine dinucleotide, that means FAD. This stands for FAD. And question is about complex 2. Okay. Option C is flavin adenine dinucleotide quinone succinate, and option D cytochrome B quinone flavin adenine dinucleotide. Any idea? Most of the questions that I have taken are from the electron transport chain because that is the major target for most of the major target of the competitive exams. That is why I have taken more questions from this electron transport chain. So any idea about this? 10th? So here, if you remember, complex 2 is also known as the succinate dehydrogenase. Okay, succinate dehydrogenase. Like how the complex 1 is called as the NADH dehydrogenase, complex 2 is termed as the succinate dehydrogenase. Okay. So option A is correct in this statement, where in the complex 2 electron transport chain, electrons are delivered from the succinate to the quinone. Okay, pool. Quinone is nothing but that Q cycle molecule. Okay, pool by the flavin adenine, and here in this process, FAD get reduced and that it produce the FADH2. Okay, here the option A is correct in this statement. Now the next question is a charged species. The unequal distribution across the membrane generates an electrical potential. The free energy change is given by which of the equation? This equation will be very much useful for you if you want to calculate the membrane potential, like if a certain electrochemical gradient is given to you, like the proton difference or the any charge concentration difference, charge molecules such as K plus Na plus, if ions differences are given in across the membrane, then if you need to calculate whether the facility whether it will facilitate the transportation of the molecule from inside to outside or outside to inside, or if you want to calculate the free energy, then this equation will help you to calculate that particular condition or to give a brief understanding about it. So the equation is very much important for you in that respect. So do you remember what is the free energy for a unequal distribution across the membrane that generates the electrical potential? How you will govern that equation for free energy change? Which is the correct equation? Any idea? Okay, here the correct equation is option B, which is delta G is equal to RT ln concentration in and divided by concentration out plus Z into Faraday multiplied by the psi, change in psi. Here, this equation, in this equation, delta G represents the change in Gibbs free energy. Then R stands for the universal gas constant. T stands for the temperature in Kelvin unit, and concentration in means inside the membrane what is the concentration of that particular ion, and concentration out, that is the concentration outside the membrane, plus Z is the charge on that particular ion. What is the charge on that particular ion? Then F stands for the Faraday constant, and delta psi is nothing but the electrical potential. So this is the governing equation to calculate the free energy change if there is an unequal distribution of the charges across the membrane. Okay, so remember this equation and it will be helpful for you assignment practice. Like I will solve one problem also, numerical problem based on this, and will teach you how to take that problem. This equation you will find in the lecture number 44, I guess, where ma'am has discussed about the membrane transport lecture 2. There you will find this equation and the derivation also, how this equation is being derived and how to utilize this equation for solving the numerical problems given in the transport of the ions. Now the next question is cytochrome BC1 complex contains which cofactor? Option A zinc and sulfur, option B iron and sulfur, option C iron and zinc, option D iron only. Cytochrome BC1. Any idea? Here the correct answer is iron and sulfur, because in cytochrome BC1 these are the main electron acceptor and all in cytochrome BC1 are the FES cluster. Okay, so if you remember the FES cluster, in that cluster normally they involve the iron and sulfur, right? So next question is Na plus glucose import is an example of option A simple diffusion, option B transport mediated by carrier protein, option C ion gradient driven active transport, or option D ABC transporter. Any guess? Hello? Any guess on Na plus glucose importer? Import? Present here?
Yes, sir. But we haven't seen the videos fully yet, so we are
Okay, okay,
waiting.
okay. No problem, no problem. Yeah, that was my concern also to the NPTEL team because they released it very late.
Yeah.
So no issue. You will you will get you just check the videos and then it will help you. Like
Okay.
okay, so correct answer here is the Na plus glucose import is the ion gradient driven because you can check here the Na plus in this import there are two components. One is Na plus, another one is glucose. So here normally cell doesn't keep Na plus inside. Okay, so to travel like whenever you see that Na plus is being involved, that means it will be a kind of active transport. Okay, so and importing glucose normally get the like glucose normally get inside the channel using the glucose channel such as because cell does not have much implication of the glucose import. So when these two are coupled, these are nothing but the example of ion gradient driven active transport. Okay. Next question is oxidative phosphorylation takes place in thylakoid, mitochondria, hemoglobin, or chloroplast.
Option B.
Option B. Yes, very good. This is mitochondria. Next question is phosphoglyceride are composed of option A unsaturated or saturated fatty acids linked with phosphate to which glycerol is attached, or unsaturated or saturated acids linked to glycerol to which phosphate is attached. Only saturated fatty acid linked to glycerol to which the phosphate is attached, or only saturated fatty acid linked to phosphate to which glycerol is attached. What is the correct statement? This is related to the membrane protein itself. Phosphoglyceride. What does this name compose? Any idea?
Option B.
Option B.
E.
Unsaturated or saturated fatty acid linked to glycerol to which phosphate is attached. Very correct. Yes. Option B is correct. Next question, non-polar amino acid are distributed mostly in the catalytic site of globular protein. Option B, surface of the membrane protein. Option C, surface of the globular protein. Option D, catalytic site of the membrane protein. Any idea? Non-polar amino acids, which does not have the polarity, are distributed mostly in the which part?
Sir, option D. I'm not sure. I'm guessing.
Option
B.
B. B for ball. B.
B. Catalytic site of membrane protein.
No, actually, these are present mostly in the surface of the membrane protein. Most
Okay.
non-polar amino acids. Okay, so option B is correct. Now coming to the one question that is numerical. In this, there are three parts of the question. As you not have gone through the lectures, so I will help you how these are solved, and also I will tell the concept behind this. Okay, so but in between, I will need your help to calculate the ln and like to give the answer related to the what is the logarithmic values and all. You just keep the calculator with you, or you can just type in the Google also. Okay, so that I will get your maximum involvement. So your question is at 25 degrees Celsius, see what all informations are needed for you. Okay, so here your question is at 25 degrees Celsius, the internal concentration of potassium is 20 millimolar and external concentration is 340 millimolar in the squid giant axon. What is squid giant axon? Axon is nothing but you are talking about the neural network, right? Neurons. There you will find axon, right? So at 25 degrees Celsius, the internal concentration of potassium is 20 millimolar and external concentration is 340 millimolar in a squid giant axon. Calculate the following. Option, your question, this has the three part of this question. First part is to calculate the membrane potential at equilibrium. Second part is minimum membrane potential that would be needed to drive the potential transport out of the cell. And third question is calculate the pH gradient that would be necessary to overcome the unfavorable transport of potassium and provide an additional 3 kilojoule per mole of energy. Okay, so the mathematical expression that I showed you, that I asked in the previous questions, that will be applicable to solve this kind of question. Wherever you are, so help me to write down that particular equation. That equation is delta G equal to. Can you recall delta G equal to?
Equals to RT ln.
RT ln. Very good. Then
ion concentration inside by ion concentration outside.
Very good. So what are ions given here? It is potassium ion, right? So K plus inside divided by K plus outside. Okay. And
plus
Z
ZF del psi.
Z F and delta psi. Delta psi, right? Very good. So here your question is, now what all things are given here? Try to find out. You have been given the temperature which will be required to you. But this temperature, remember this, because in gas constant you use joule per Kelvin per mole. So your temperature, you need to convert this to Kelvin. First thing. Then second thing, what is Faraday? Faraday constant here. Faraday constant is nothing but some constant. C stands for Coulomb per mole. Okay. And then Z stands for the what is the charge on that particular ion, right? Now psi is nothing but your membrane potential, which you need to calculate. Okay. Now the trick or the concept that you need to recall is at the equilibrium. At the equilibrium means the reaction is not going, means there is no transport from inside or outside. So at equilibrium, what will be delta G? At equilibrium, delta G should be 0. That is why it means it is not spontaneous nor non-spontaneous. That means the membrane is static, right? There is no transport, no import, no export. Okay, so means free change in the Gibbs free energy will be 0. So that means RT ln K plus in by K plus out plus Z F delta psi will be equal to 0. Okay, this is your first thing that you need to do after seeing that there is a equilibrium. Okay, now let me convert all the essential thing. Your temperature is given is T is equal to 25 degrees Celsius. You need to convert this into Kelvin. So what you will add? You will add 273, right? 273.15. 273.15. It will convert 25 plus 273.5. That means 298.15 Kelvin or 298 Kelvin, right? Now your temperature is getting converted. Next thing, you have 20 millimolar and 340 millimolar. Okay, so this implies you now just multiply. R is universal gas constant, so 8.314 multiplied by T, which is 298. Then ln inside is 20 millimolar divided by outside is 340 millimolar. plus Z. Z is what is the charge on potassium ion is 1, right? So Z will be 1 into Faraday constant 96485. Okay, multiplied by delta psi, and you need to calculate delta psi only, and this is equal to total is equal to 0. Now you put this that side, like in you keep this right hand side, so it will give you delta psi is equal to, I will just keep shortcut, delta psi is equal to 8.314 into 298 divided by 1 into this 96485 into ln. Now remember one thing. Here you have taken this particular term to right hand side. It will convert the sign to minus, right? And then that minus you can use to like reverse this ln thing, right? So that will become, if you are taking this as positive, then it will convert to ln 340 divided by 20. Now can you quickly use the calculator and calculate what will be the value for this? What will be the membrane potential at equilibrium? Or I will use the calculator and then we'll go to the calculator. Okay, so here you have 8.314 multiplied by what is this? 8.314 multiplied by 298. Then this particular thing divided by 1 into how much? 1 into 9896485, right? This will give you 0.2 this particular thing, and then you multiply this by ln. Find out where is the ln, and that you will divide here 340 divided by 20. 340 divided by 20, and this will give you the 0.0727. Okay. Is there anything wrong? 20 millimolar. Yeah. So, but your concentration will be per mole. That means you need to multiply this particular value into 1000, and that will give you how much. This is particular volt, right? Delta psi you have calculated in the volt. Okay. The value you have got is. Let me take the pen access. So here this will give you the 0.0727 volt, and if you convert this into millivolt, that will give you the 72.7 millivolt. Now you are exporting. Now you are calculating the membrane potential. That is mostly because you have taken this as psi. So membrane potential is mostly in the negative value. So delta psi will be minus 72.7 millivolt. So your first part is solved. Now your next question is to calculate the minimum membrane potential that would be needed to drive the potassium transport out of the cell. Okay, so in the next part, what you will be required, you need to, the temperature is same. Recall everything. Temperature 25 degrees Celsius means 298 Kelvin, right? Then you have internal, K plus internal is K plus internal is how much? 20 millimolar. Sorry. Yeah. Then K plus external. This will be your what was the value? 340. 340 millimolar. And Faraday constant, these are all given to you. Now you need to calculate what will be your, now you need to calculate minimum membrane potential that would be needed to derive the potassium transport out of the cell. Okay, so out of the cell, if you want to calculate, that means you need to calculate what value? Delta G, chemical potential, right? So in that, you will need the same thing. Minimum means that also represents the value at which the equilibrium, like just beyond the equilibrium, right? Minimum potential is talking about nothing but at which the first transport will happen. Means if you increase that particular thing, like if you exceed that particular membrane potential, the transport will happen. That means if you calculate the at equilibrium itself, that will be more than enough for you. So here, if I draw a membrane, so this is a particular membrane. So inside the membrane, you have K external, K internal. Sorry, inside the membrane you have this, and outside the box you have K external. Okay. So now if you calculate the delta E value for this facilitating this one, so that will be also the same. Delta E is equal to RT. So delta E will be RT divided by ZF ln C external. Why external? Because now you have to change the sign, right? C external divided by C internal. And this will also give you the same result, right? So you will get the same thing which will be minus 72.7 millivolt. Okay. What was your next question? Your next question was to calculate the pH gradient, which would be necessary to overcome the unfavorable transport of K plus and provide an additional 3 kilojoule per mole of energy. Okay, now you need to calculate three different things. Here, first you need to calculate what is the pH gradient. Okay, and then you need to get the additional energy of 3 kilojoule per mole after this unfavorable transport. You need an extra 3 kilojoule per mole that you will need to incorporate in your equation. Okay, now the same equation will result you the same equation, which is delta G is equal to RT ln. So here RT ln, okay, K inside bata K plus outside. This is for your delta G. So if you are not including the membrane potential, then you need not to add the Z plus term. Okay. If you are adding that particular membrane potential, then only you need to add that ZF delta psi, because here you have been asked only regarding to the K plus and only regarding the pH gradient that would be overcome for unfavorable transport of K plus that provide the 3 kilojoule per mole. So in that case, you only need to take this equation, delta G is equal to RT ln concentration, which is the main equation of delta G, gives free energy, right? So if you overcross this particular energy, then you will be able to transport the molecule from one side to another side. So delta G will be RT ln. R is nothing but 8.314 into 298 ln K plus inside is 20 millimolar divided by 340 millimolar, right? And this will give you, let me calculate this. Okay, so 8.314 multiplied by how much? 8.314 multiplied by 298. Then again multiplied by ln 20 divided by 340, and this will give you something about 27091. Okay, this much. This is your Gibbs free energy. Means 7019. Okay, this was your answer, right? This is you calculated 7019.49. This is joule, because everything, all units were in joule. So joule per Kelvin per mole. And here the Kelvin has been cut. So joule per mole, right? So this much joule per mole, and there's delta G means difference is there. Now you need 3000 more kilojoule per mole, right? 3 kilojoule per mole means 3000 joule per mole. The extra energy will be delta G plus 3 kilojoule per mole, and that will be equal to your proton gradient, right? Your question is to calculate the pH gradient. So in pH gradient, what will be there? There will be a proton difference, right? So that you will equate to RT ln, same thing. RT ln change of ln H plus out H plus in by H plus out, right? Okay, and here you will get this 7019 plus 3000. Both will be in the negative side only, because 3000 extra means you will add this one. So it will give you the 10,019.49 joule per mole equal to RT 8.314 multiplied by 298 ln of H plus out. Okay. Oh, sorry, H plus in divided by H plus out. And from there, you will get the ratio of H plus by H plus out, right? I will just tell you how to do that. You will calculate the H plus in by H plus out. And it will give you some value, some C. Okay. Then you take, then you simplified this by H plus in equal to C into H plus out. I'm just telling you the steps that you can follow. H plus out, and then you take logarithmic log, because pH is nothing but log of H plus, right? So you take log of H plus in both sides. That will be log C plus log H plus out. Okay, this will give you the minus pH of in, pH inside is equal to log of some value which you will get. You calculate the log of that and then you will get that value. You term that as K plus some. This will be minus, because pH is equal to minus log H plus, right? So this will be pH out. So here, if you take the difference of these two, that means it will give you the K value. This K will be the difference between the pH, pH gradient. You calculate the pH gradient. This will be the pH gradient, which would be necessary to overcome the unfavorable transport of K plus, and also with the addition of 3 kilojoule per mole of energy. Okay. By this, I told you about solving these all three kind of problems, and then now I will take any question if you have. Are there any question or doubt related to the solving this problem, or any regarding to any problem you faced? Any doubt? Andrea, Swarnava?
No, sir, I have nothing.
Okay. Andrea?
No, sir.
Okay. Okay. Then we will move ahead with the like that thing I wanted to share with you regarding to the animation video. So this and this time I will this time I will do the caption on, so that you will be able to read the comment if you are not able to see the, like if you are not able to listen the video. So I will start this. This is regarding to the endosymbiosis. Endosymbiosis means like now the eukaryote, like eukaryotes have them, like all the eukaryotes have mitochondria in them, which facilitate the this one, like uh, energy generation. So how this got inside the cell? Because mitochondria was nothing but it was previously it acted as an independent organism, which is a kind of a prokaryote because it has its own genetic material, DNA and all. So you just watch this video, it will help you to understand that particular concept. Read the captions. So this was all about how the mitochondrion get engulfed in the eukaryotic cells and how these two independent cells cooperated and evolutions exist. Like if that process didn't happen in the past, then this the fate would not be as much happier that we have today. Otherwise, like humans have not been evolved out of that. Because of this symbiosis relationship only, the bigger cells which are eukaryotic would able to harness the potential the oxygen thing energy from oxygen using the help of mitochondria, which again helped the mitochondria also because the larger cells were able to give the energy sources to mitochondria, and then this symbiosis was helpful for both of the cells. So this was about how mitochondria got engulfed in the eukaryotic. Next thing is why mitochondria is called the organelle of the cell that are formed. Why it is called the powerhouse. This particular animation is regarding to that. This is the upper one is the outer mitochondrial membrane. This is inner mitochondrial membrane, and in between it is the intermembrane space. Okay, they are giving the pictorial view of the this particular mitochondrial structure. Now they will describe about the different complexes that are involved in the electron transport chain. The electron transport chain will be again explained in the later video, but first let get a overview of these complexes. So now you understood the why mitochondria is called the powerhouse of the cell, because it involved the different like on the membrane of the inner mitochondrial membrane. It caters all the complexes that are essential for the electron transport chain, right? And you also got a knowledge about the how these mitochondrial membrane are being strategically located around the cells where the ATP ADP concentration is being maintained, where the energy is most needed, right? And these are traveled through the microtubules that you studied in the last class. And now the question is how the mitochondria travels across the microtubule with the help of which motor protein? Can you answer this question? Which motor protein travels across the microtubules? Can I get answer? If you remember correctly, it was kinesin and dynein, right? These two motor proteins travels on the microtubules. However, the different myosin travel on the particular different microtubules, different structure, right? So now the next video is about like how the electron transport chain, different complexes and ATP synthase is located on the inner mitochondrial membrane. So let's start the video. Please read the caption. That will be very So by now you know why different complexes are important. Okay. So I will paste this video again in the forum along with the recording of this. Like when I will post this recording into the YouTube channel. And also, if there are any doubt, you can ask me now and also in the discussion forum. So I hope you enjoyed the video and this lecture was beneficial for you. You cover the lectures that are uploaded in the NPTEL, and if there are any doubt, you can watch the recording of this like a live interaction session. Okay. Are there any doubt, Andrea? Hello? I cannot hear you.
No, sir, there are no doubts.
Okay, then. Thank you so much for joining. We will end the session now. Thank you.
Okay, sir. Thank you.